In this video, we're going to talk about muscle tension, or the amount of force that a muscle actually generates. We're going to talk about some factors that affect how strongly a muscle can contract and how quickly a muscle can contract. The first thing to consider is what's called the length-tension relationship. And the idea here is that how strongly a muscle can contract depends on how long the sarcomeres are within the muscle before this contraction actually begins. So let's take a look at the diagram to the right. So the first thing that I want to talk about, of course, is what the y-axis means, and that is the tension that's actually developed as a percent of the maximum possible tension. So of course we're just going from 0 to 100 here. And on the x-axis you have the resting sarcomere length. So at what length is the sarcomere? So I want to kind of remind you, think about your biceps brachii and think about the fact that it can be relaxed whether your arm is bent or whether your arm is extended. So basically the position that the biceps begins in is going to determine the force with which it can contract. Now the example that I kind of want to use here for overall tension is I want you to think about this. As you do a bar curl, it's hard to get the movement started when you are lifting at just about your maximum weight. Difficult to get that movement started, and up at the very top of the muscle, it's difficult to finish that movement out. So a lot of the time when you're lifting weights, difficult to start, difficult to finish, and we're going to find out exactly why that is over the next couple of slides. So if the resting sarcomere length is really short, let's say that your limb is already flexed before you begin to contract it. Below a sarcomere length of about 2 micrometers per sarcomere, you have a very large zone of overlap. And so this is the point at which the thick and the thin fibers actually overlap. Now you might think that more overlap is a good thing because you have a lot of cross-bridging available. But what actually happens when the sarcomeres start out short, very quickly the thick filaments here actually get compressed by the Z-discs here as the Z-discs move in towards the center of the sarcomere. So what this causes is crumpling of the thick filaments, and the myosin heads get out of alignment. And if you'll recall one of the earlier diagrams in another slide, you can see here that the orientation of the myosin heads is going to be pretty important in determining whether they can actually bind with the actin fibers on the thin filaments next to them. So with this orientation being important, imagine that the myosin heads you see here are kicked off to the right or the left, or are out of position. That means that even though the myosin might approach the actin, it will hit on the active spot or on the active site. So basically, with fewer cross bridges because of misalignment, the overall tension generated by your muscle is actually weaker. Okay, and so what this diagram shows you is at about 1.8 micrometers. So this original diagram that we just saw. Okay, so let's move to the right and let's make our sarcomere length a little bit longer. So now we're going to actually lengthen our sarcomere out a little bit. So at a sarcomere length of about 2.0 to 2.4, this is actually close to the resting length in most muscles. At this point you have some overlap, but not as extensive an overlap as you saw in the previous slide. So in this case, the zone of overlap, as you can see here, is from the edge of the H zone, and remember that the H zone is where you have thick filaments only. So the edge of this H zone to the end of a thick filament, and that's where this bracket is, this is your zone of overlap, and you get maximum tension here. So because there is a lot of cross-bridging occurring, and none of the myosin heads are misaligned, you've got the maximum number of cross bridges and can therefore exert the maximum amount of tension. Now I do want to point out, as you can see in the graph to the right, that there is a range to the optimal length. And so the optimal length actually occurs in that gray area, and I'm sorry, it's a little bit misaligned in here. The optimal length occurs in that gray area where you can generate the maximum amount of tension from about 80% of resting length to about 120% of resting length. Okay, so let's stretch our sarcomere out even more. And in this case, we're going to go to a little bit of the extreme and stretch it out to 170% of its optimal length. This is at about 3.8 micrometers. Now, in this case, as you can see, there's actually no overlap between thick and thin filaments. If you look at a little diagram just below the first paragraph, no overlap between thick and thin filaments. And because there's no overlap, cross-bridging is actually impossible. The muscle fiber can't contract. The tension here is actually zero. And so by the time you get out to this resting sarcomere length, by the time you get stretched out to this point, you have what's called a strain, which occurs when the muscle fibers tear because of overstretching. This is something that's also commonly known as a pulled muscle. So whenever you pull a muscle, it just basically means that the sarcomeres in that muscle have been stretched out further than they can actually bear. Since we mentioned muscle strains, let's talk about how they heal. So when you tear muscle fibers, initially when they're torn, they'll die off a little bit, but generally only for a short distance, and fortunately not the entire length of the muscle fiber. All right, the very first thing that happens is that satellite cells start to proliferate, so they start to carry out cell division. Quite some time ago in a previous video, I told you that we would examine the role of satellite cells, and here's a role of them. They're very important in terms of healing muscle strains. So what they're going to do is they're actually going to differentiate into myoblasts, and if you'll recall, these are the cells that actually join together to form the muscle fibers in the first place. So they'll join together, they'll form what are called myotubes that are multinucleated, and then basically what will happen is that they'll fuse with the injured muscle fiber that survived the trauma. Now, sometimes muscle tears as a result of overstretching are so extensive that they actually kill the cells. At that point, there's no coming back from that. You're just going to get scar tissue formation. So let's talk about muscle strain healing. Now, the first thing again that happens is that the satellite cells proliferate in response to the injury. Okay, so if you look at this diagram, you can see where the muscle fiber has actually been torn, and those little things in the middle are satellite cells that are proliferating in response to the injury. The next thing that happens, you see the little star-like objects there. The satellite cells are differentiating into myoblasts, and then here they start to join together to form multinucleated myotubes. The next thing that happens after this is that they'll fuse with the injured myofiber that actually survived the trauma. Okay. Now, if you look at this diagram, you'll also see it about the same time. So you see the myotubes there with the multinucleated myotubes on the fourth diagram down there, but you'll also see in the middle here that you're producing a connective tissue scar. Okay. The next thing that happens is that the myofibers that actually survived will at least slightly join with the myotubes, and they'll try to actually pierce through the scar on either side. So you can see here that they're reaching out these sort of extensions, and these branches actually adhere to the scar. So the problem that happens here is that when those branches actually adhere to the scar, it makes this myofiber a little bit stronger than the adjacent cells. So the problem here is that they're more susceptible to injury if they're re-aggravated. And again, one of the problems that you can run into is that by the time this happens, when the branches are adhering to the scar, you're not going to have a lot of swelling or pain at this point, and you're going to feel like you can work the muscle as well as you normally could. And so the unfortunate thing is that the muscle is still quite weakened at this point, at this injury point. And so you've probably seen athletes coming back from pulled muscles. Usually pulled hamstrings are a real common theme in football and in basketball and in soccer. And so when athletes try to come back from a hamstring pull too quickly, it's very common for them to re-injure the muscle at the same place. And in this case, because the point of injury is weaker, then you end up with a more extensive injury than what you had originally. So if you ever pull a muscle or have a muscle strain, it's always best to try to keep from working that muscle too hard for some time after you feel just fine. So the reason why you want to stay off that muscle for a little bit longer is because this allows, there's not a picture of this, but this actually allows the scar to diminish, and once the stumps come together and those myofibers become interlaced, then you're going to have a muscle fiber that's approximately the same strength as the surrounding muscle fibers. So one thing that I should say here is that the scar tissue is pretty persistent, and how much of a function you recover of the muscle really does depend on the extent of the original injury. So that is definitely something that you have to see. So let's say you have a slight hamstring pull during an exercise, and 3 or 4 weeks later you're able to get back on it, and you don't notice a decrease in performance. That's because you've had a minor injury. I've seen videos of people who are trying to bench press too much weight, and they tear. They have these terrible tears of the pectoralis major muscles, and when that happens, the tearing is so extensive, the scar tissue that stays persistently in there is so extensive that they never recover the full capacity of that muscle again. Okay, so how is muscle tension actually controlled? So there's a new term that I want to mention here, and that's called a motor unit. And a motor unit is a motor neuron. This is a neuron that directs actions, so it'll carry instructions from the central nervous system to an effector, in this case the skeletal muscles. A motor unit is the neuron and the muscle fibers that it innervates. So one thing to say about motor neurons and neurons in general is that they are able to branch very extensively without the intensity of their action potential diminishing. So basically what that means is that you can have a neuron that can branch once or twice or actually several hundred times, and if an action potential starts at the axon hillock or right close to the beginning of the neuron, then even if there are hundreds of branches, each individual branch will receive the same intensity action potential, which means that each of the muscle fibers that are associated with this one neuron get the same intensity signal. So one other thing to say is that this is not the same as a motor end plate. The motor end plate, if you'll look here at the diagram, you see where the little circles are in the green and purple lines. So the green and purple lines represent neurons, and the little circles represent axon terminals. There's a motor end plate beneath each one of those circles. And so a motor end plate is just the area on a muscle fiber that can receive neurotransmitters. So a motor unit, on the other hand, is one motor neuron and the muscle fibers that it innervates. So as you can see here, even if you're just touching one neuron, there can be multiple motor end plates in one muscle fiber, much less several hundred muscle fibers. Okay, so these motor units, if you have precise movements, they're going to have lots of small motor units. So muscles of the larynx that control voice production, they'll only have 2 or 3 muscle fibers per motor unit, and this results in a lot of precision. So if you think about voice production, the tension that's generated on the vocal cords is what causes changes in pitch. The different sounds that you make as you talk are really controlled by the muscles of the mouth, but also the position of the larynx as you talk. So one thing that I'm not doing, and that I really try not to do, is to talk in a monotone. And so if someone is talking in a monotone, it really means that there isn't a lot of movement of the larynx or the muscle surrounding it. I'm not going to do that to you for too long. So as your voice goes up and down in pitch, the position of your larynx is changing. Moving your eyes takes a great deal of precision. So on the many occasions where you have rolled your eyes at the jokes that I've made in class, your eyeballs have followed a very specific track. And so the muscles that control that eye movement can have 10 to 20 muscle fibers per motor unit. So that's still not very much. And because there aren't a lot of muscle fibers involved, there's precision, but there's not a great deal of strength. Now, the other side of the scale is skeletal muscles that have to carry out large scale movements and powerful movements. So the biceps brachii in the arm, which is pictured here, and the gastrocnemius, which is the calf muscle in the leg, these can have 2,000 to 3,000 muscle fibers in some of their motor units. Now, because when neurons branch, again, they send an equivalent action potential at the same time to each of the cells in the motor unit. So because when all of these muscle fibers in a motor unit contract and relax at the same time, the strength of a contraction depends on how big the individual motor unit is and how many motor units are actually activated at a given time. So one thing that I want to make clear is that in a muscle like the biceps or the gastrocnemius, there are a great deal more than 2,000 to 3,000 muscle fibers in these muscles. So you're going to have the biceps made up of a number of motor units. You're going to have the gastrocnemius made up of a number of motor units. But so how strong is your contraction? It depends on how big the motor units are and how many of them get activated at the same time, because even within the same muscle, you don't see always simultaneous activation of all motor units.